Thank you very much, and thank you for attending. I'm looking forward to telling you about Life Edit, so I can see some familiar faces, so this is lovely. To start with, Life Edit is a wholly owned subsidiary of Elevate Bio, and we are focused on gene editing. We've built a platform that is both broad and deep, that we have developed to allow us to really be able to edit anywhere in the genome that we want to be able to do an edit. And we've built the technology so we can do the specific type of edit we want to do, and I'll talk about that in some of the later slides. We're using this platform internally to develop a an in vivo gene-edited therapeutics pipeline, and we are actively partnering we actively partner the platform for enablement of cell therapy and for specific therapeutic areas for in vivo. So what is this platform? Where did it come from? We actually have access to a proprietary collection of microbes that's been fully sequenced through a company called AgBiome. We're based in RTP in North Carolina. AgBiome is there, and that was kind of the company from which we came. Upon mining that collection, we've been able to find hundreds of thousands of nucleases, and you see some representation of that on the right-hand side of the slide. What was fascinating about this collection was the amount of PAM diversity that we were able to identify. And so we do a lot of this bioinformatically, and then we take it into the wet lab, we characterize it, and we try to understand the kind of editing capability of these nucleases as they come out of the system. We then make decisions about which ones we want to take forward. Some of them, we make the decision based on size. Obviously, we think about IP, and we think about our ability to have the broadest PAM diversity with the smallest number of nucleases. So at this point, we've identified, or we have focused on, I guess would be a better way to say it, on 4 of our nucleases. We call them LEGs. It stands for Life Edit Gene. So 98, 14, 145, and 95. And these have a couple of, we have chosen these nucleases for a number of reasons. So first of all, with those PAMs, we've got a pretty orthogonal set of nucleases that we can use as the foundation for anything we want to do with gene editing. They're all relatively small versus SpikeCas9, so not as small as some of the type 5s out there, but close to 1,000 amino acids, and that really allows us to package them into AAV if we want to, along with guides and the regulatory elements required. And we've done that, and we've actually done it when we've got these attached to base editors as well. These have low homology to the established systems. We have a great IP portfolio and IP strategy, and so we're very comfortable in the way we're moving these forward. So those nucleases themselves are kind of the foundation of the gene editing platform that we're building. And we use them obviously for double-stranded breaks and knockout. We can use them for insertion and repair, and I'll show you some data a little later in the slide deck. But we've also used them as the kind of the chassis for a base editing platform that we have developed using proprietary deaminases that we found in the same collection. And for RT editing, which we're beginning to build now within Life Edit. So the decision to go after base editing was made pretty early on, and it was made because we understood that the power of the PAM diversity was going to be most important with base editing, because you've got such a small window in which you can make your edit. And so we've really developed both C and A base editors. They're modular, they're proprietary, so you can take the deaminase, the kind of a selection of our deaminases against a selection of our nucleases and develop the editor that you need for the specific thing you're addressing. They're efficient, they're specific, we don't see double-stranded breaks, low indel formation. And so we start to think about how do we want to use these base editors. And so they do have, different uses. So with the C-base editors, we can introduce stop codons, and we can do disruption of splice sites. They can correct disease-causing SNPs, but they require a USP as well. And so we really think about the C-base editors as having their biggest role in cell therapy and thinking about multiplexed editing in that space. And I'll show some data there. Our A-base editors, we think about them more in the context of in vivo gene editing. We predominantly using them for disruption of splice sites and thinking about utilizing them to correct disease-causing SNPs. Obviously, we could use A-base editors to enable cell therapies, but that's kind of not been our focus up to now. So I thought I would talk most about how we're thinking about these in the ex vivo cell therapy space. Obviously, if anybody's interested in hearing more about our in vivo therapeutics or how we're delivering them, we can have that conversation offline. Most of this is being done in conjunction with either Elevate Bio or external partners. And we are at the point now where we've got really editing across a very large number of cell types. And we have started to build a platform for knockout and knock-in using multiplex base editing that we think is going to be pretty modular, where there'll be a kind of baseline of edits that we feel that a lot of people are trying to use and be a little bit off the shelf. And then we can add in bespoke edits, bespoke cargo that you're knocking in. So we'll touch on that as well. So this is knockout data, and just showing very quickly from left to right. On the left-hand side, we show very, very high levels of editing. This is both mRNA and RNP. This is using leg14, and you can see that we are getting highly efficient knockout across a variety of different cell lines. The middle panel is showing that you have a target gene that you want to knock out. We can pretty effectively and efficiently design a set of guides and then screen to understand which guides are going to be most applicable for your edit of choice. We can nearly always find a guide. Well, not nearly always. We always find a guide and that will do this efficiently. And then next to that, obviously, you're not just looking at your editing efficiency. It's got to be doing something functional. And so we use a variety of techniques to show the effect on the protein. And then finally, Elevate Bio has a strong regenerative medicine team, so a strong expertise in iPSCs. They leverage our gene editing platform, and they're doing a lot of work to show the editing in the iPSCs. And you can see here an example where we've done knockout and very, very efficient. So that's knockout, knock-in. So we've done a variety of work in this space using AAV to deliver a number of different cargoes. And again, we're seeing very high efficiency of the knock-in. In fact, I think if our CSO was here, he would say something along the lines of, this is some of the best data that you'll see out there. And we've done this repeatedly. This isn't a one-off. There's nothing special about those TCRs, and we've looked pretty closely. And so when we started to see how robust this knock-in data was, we started to recognize that there might be an opportunity to build something useful, not only for ourselves, but for others. So let's just talk about the C-base editing and why you might want to multiplex. So if you're going to do, or not why you might want to multiplex, why you might want to use C-base editors to do your multiplexing versus a nuclease. So obviously, if you're doing a single edit, using a nuclease, doing a double-stranded break might be just the right way to go, and that's great. But as you start to want to do more and more edits, as you do more and more of those double-stranded breaks, you're going to have complexities in terms of translocations and what have you. Your off-target, your opportunity for off-targets is going to get much higher. And so we think using the C-base editors is a better approach. And so we've started to build some data to show that we can do this and we can do it efficiently. And then after showing the efficiency through NGS, we start to look functionally and see that the editing's doing what we expect it to do. And so just walking through this slide, so under the red number 1, this is really the initial singleplex gene editing data. So this is taking a single nuclease with each guide, and you see the efficiency of the knockout at each of those targets. And so, we've got reasonably efficient knockout across these genes, and the protein knockdown has been verified with flow cytometry in the Western blot. So then, having gotten that data, we then took it and asked the question, what happens if you tried to do this together? And so under 2, you see now a 5-plex where we're editing the same targets with that nuclease and the 5 guides. And yes, we do get a drop in efficiency, but you're seeing, between 60 and 80% editing across all those targets, all those genes. And then most importantly, when you look at the protein knockdown, we're seeing what we would expect to see. And in fact, and this is not unexpected, the percent of protein knockdown looks greater than the kind of percent editing that you're seeing. And so these now are going forward. These are part of a larger program within Elevate Bio. So the functional testing of these edited cells is now being evaluated in vivo. And as we get that data, we'll start to share it. So we've got we're always thinking about what can we do with this broad set of tools that's, interesting, useful, that others might be, thinking about what problems can we solve. So we've done the multiplex base editing. We also recognize we've got this strong knock-in data. And then because we have this PAM diversity, we've got multiple tools. So we can do multiple edits at different locations because of the access to the different PAMs. And so, as I mentioned earlier, we started to think about building this, we call it knock-in, knockout. I think it should be knockout, knock-in platform, where we combine the nuclease and the base editor to be able to more precisely engineer cell therapies and do it in one pot, right? So what you don't want to do, I suspect, is do one edit, pull your cells out, and then do the next edit. And so we wanted to come up with a way where this could be done efficiently in a single pot, in a time frame that's reasonably aligned with what you would want for a cell therapy. And so what we've been able to do is use a nuclease with the track guide to do the double-stranded break, into which we're able to insert a TCR of interest using AAV in this case, and at the same time do the knockout of B2M with a C-base editor. And so because they have different PAMs, they're going to different parts of the gene. And it's all done with a single transfection. So this is an initial proof of concept. There's some optimization to be done here, but this is all moving in the right direction. So again, looking at the slide from left to right, in the first slide you get the control. In the first panel you see the control. In the second panel you see the knockout of B2M, which is removing then the endogenous TCR. You see the knock-in of the TCR as you move to the third panel. And then in the final panel, you see both the knock-in and knockout. So about the third of the cells had the correct TCR and the B2M knockout. We're working to optimize that. We're very confident we'll be able to get this significantly improved. And we think that this offers an opportunity to build a broader platform where we can offer certain knockouts, that are already predefined. We've got the conditions, we've got the gene editing in place, and then we can work with partners to do the knock-in that they're looking for, or the bespoke knockout maybe that you want for your product. Doing all of this is great. We hope all the on-target goes well. We're also thinking about what's the off-target strategy to really understand what's going on. And, again, this is very dependent on what you're doing. So we, very early on, in the process, even when we're doing the early guide design, we start to think about what did the guide, what's this going to look like bioinformatically? And then we run, when we do the initial screening of the guides, we also run the amplicon-seq to look at the off-targets there. We're using digenome-seq, and then as you move through, as you're developing your product further along, we start to think of some of the other assays that we'd want to bring to bear to provide confidence that you understand what your off-target profile is. And then finally, one of the things that we've really thought a lot about is, if we're working with partners, how are we supplying the gene editing composition? And so we've really worked to provide a secure supply of these gene editing compositions in a number of different formats. So with RMP, as mRNA, we can provide that to our partners as they need it. If it's going to be delivered as an AAV, Basecamp at Elevate Bio can provide that material. And obviously, we'd be excited to talk to somebody who was interested in doing the gene editing and to have Elevate Bio do the cell manufacturing. And that, I believe, is it.